Hello, everyone, and welcome to the second session in our T-Cell Talks event. I'm Andrew Davies from the events team here at scientist.com, and I'll be your host for this session. Joining us are Dr. Amanda Durkin and Dr. Fran Antoniano, respectively product manager in immunology and principal scientist in research and development at STEMCELL Technologies. They will showcase STEMCELL's T-cell isolation solutions, focusing on large-scale research strategies. Amanda will first give a presentation, and Fran will join us as well for the Q&A. With that, I'm pleased to welcome Dr. Amanda Durkin. And Amanda, it's great to have you with us. Feel free to take it away whenever you're ready.
Awesome. Thank you, Andrew. Hi, everyone. I am Amanda Durkin. I am a product manager on the immunology team here at STEMCELL. And I'm going to be talking about some of the next generation cell isolation solutions that we offer. As many of you probably already know, STEMCELL Technologies is a global biotech leader, and we've been developing innovative, high-quality reagents and tools for over 25 years to support many different areas of life science research. What we're going to focus on today is some of the support that STEMCELL provides for the field of immunology research. And although we offer products for your entire workflow, this presentation will be focused on T cells. So to start, what are T cells? In the simplest definition, T cells are lymphocytes that have matured in the thymus. T cells express a T cell receptor that will interact with a specific antigen signal displayed on major histocompatibility complexes throughout the body. Once the T cell interacts with its cognate antigen and co-stimulatory signals, the T cell will become activated and carry out specific effector functions. There are two main types of T cells in the body: CD8 positive cytotoxic T cells and CD4 positive helper T cells. For a human-specific context, T cells comprise of about 10 to 20% of leukocytes. So what are the signals necessary to activate a T cell? There are three types of signals that will activate and instruct the T cell on the required effector function. Signal 1 on the far left here is the T cell receptor interaction with the antigen displayed on MHC by an antigen-presenting cell. This interaction informs the T cell that it is responding to its cognate antigen. Without additional signals, the T cell will become anergic and no longer become activated. Signal 2, in the middle of the slide, is a co-stimulatory signal provided by the antigen-presenting cell that will result in the cross-linking of CD28, and this tells the T cell that the antigen being responded to is being presented by an appropriate cell type, and that activation in this case is warranted. For signal 3 on the far right, further instructions can be provided to the T cell by various cytokines secreted by both antigen-presenting cells or other cells. So in total, the influence from all three signals will result in a tailored response to the pathogen being encountered. So depending on the specific signals received by the T cell, various effector functions can be undertaken by T cells. There's direct cell killing, which is an outcome of CD8 positive cytotoxic T cells becoming activated, whereas CD4 positive T helper cells will secrete specific cytokines in response to that particular pathogen that was encountered, which will further modify the response against that pathogen. Another hallmark of T cells is their immunological memory of pathogens encountered, which allows for rapid clonal expansion upon future exposures to pathogens. Lastly, T cells can actually work to suppress other immune cells, for example, regulatory T cells that suppress autoreactive immune cells, which maintain peripheral tolerance. So you might imagine with the wide variety of functions for T cells, there are a lot of applications for their use. T cells are used in research areas ranging from fundamental biology research, cellular therapy development, modeling of diseases, and discovering new drugs. Harnessing the power of T cells for these applications is incredibly important to move this important field forward. In the rest of this presentation, we will highlight some of the key areas that stem cell products contribute to these workflows. Specifically for me, though, I'm going to be talking about our cell isolation products. So here at STEMCELL, we've worked hard over the years to ensure that the products we develop support every step of immune cell-based research, from sourcing of primary cells to cell isolation, activation, genome editing, cell expansion, and analysis. As I mentioned, today I'll be focusing on our cell isolation products. And in order to provide a foundation for the next portion of my presentation, I will first introduce you to our existing cell isolation technologies, particularly our immunomagnetic approaches with EasySep. So for those who may not be familiar with EasySep, this technology combines the specificity of monoclonal antibodies with the simplicity of a column-free magnetic system. The result is an incredibly fast and easy protocol for the isolation of highly purified immune cell populations. As you can see here, the protocol really is as simple as adding EasySep reagents, which is the antibody cocktail and magnetic particles, to your cell sample, incubating your labeled sample in an EasySep magnet, and then simply pouring or pipetting off the supernatant to separate the labeled and unlabeled cell fractions. The EasySep portfolio that we've developed over the last 20 plus years has been used by thousands of researchers worldwide and works robustly across a wide variety of species and starting samples with different selection methods and magnetic platforms available depending on the sample volume and the number of samples that you need to process. These magnetic platforms here are fantastic for processing relatively small blood samples, but up until recently, there simply hasn't been a user-friendly way to process much larger samples. And there's definitely an increasing number of applications where processing large sample volumes is not only quite common, but actually a critical part of the workflow, primarily in biobanking, drug discovery, and immune cell therapy research. In these areas, the use of primary immune cells is an incredibly important tool for ensuring the physiological relevance of the research being conducted. In these areas, the use of these primary human immune cells is incredibly important. And compared to cell lines, primary cells more accurately reflect inherent variability in donors and samples, which in these cases is actually a good thing. This allows them to facilitate translation of basic research into preclinical and eventually clinical applications. And leukophoresis samples, or leukopaks in particular, are an excellent sample source as they can provide large numbers of single donor MNCs from peripheral blood to ensure enough cells are available for use across a multitude of downstream assays. While many groups perform preliminary work with smaller sample volumes, most in these research areas eventually need to scale up to larger volume samples like leukopaks. But efficiently isolating cells from an entire leukopak can be tricky, and there are some considerations unique to processing them, namely ease of use, protocol speed, performance, and of course, really easy integration with existing downstream applications. So with those considerations in mind, this is what a typical large volume processing workflow can look like. Starting on the left, a leukopak can contain 100 to 200 mils, or even more, and about 9 billion cells. When processing large volume samples, the first step involves processing that large volume sample into a cell suspension, typically with washing. And the method you choose to prepare the cell suspension will depend on the cell type you want to isolate. However, given the capacities of most cell separation systems, the samples need to be split prior to processing, as most systems are designed and really optimized for use with far smaller volumes of blood, typically 1 to at most 50 mils. So in order to process this large volume, you need to split that sample and perform multiple small-scale cell isolation procedures, after which you end with your cells that are ready for downstream applications. But a really good system for processing leukopaks needs to be easy to use with those larger volumes. So now with a large-scale cell isolation platform, you can take that cell suspension and with no splitting of the sample, perform a single cell isolation procedure. By eliminating that need to split samples, it increases efficiency, as protocol speed can be a really big factor when working with these large volumes. So with just a single cell isolation procedure for the entire leukopak, Being able to process up to 225 milliliters or 12.5 billion cells per separation procedure is very efficient. And most importantly, the system needs to deliver the same highly purified functional cells that you were obtaining previously at the smaller scale to ensure that the results of your downstream applications translate appropriately as your research scales and progresses. So overall, large-scale cell isolation platforms require a fewer number of steps to isolate cells from a large volume sample compared to that small-scale cell isolation platforms. With that being said, I want to now introduce you to our new large-scale cell isolation platform. So this is our EZ250 EasySep Magnet, and I'm happy to highlight that it is the newest addition to our manual magnet portfolio. The EZ250 EasySep Magnet is designed to isolate cells from large volume human whole blood and leukophoresis samples in the range to 25 to 225 milliliters, or up to 10 billion cells, which represents a typical full leukopak. Depending on our reagent offerings, we have different volume limitations between our negative or positive selection kits. But just like our other EasySep Magnet platforms, the EZ250 protocols are incredibly fast. Typically, they can be completed within 20 to 40 minutes. This means that you can process a full leukopak in roughly half an hour in a single experiment without needing to split your sample across multiple tubes or magnets. This magnet uses a standard T75 cell culture flask, and we have a variety of large format reagents and optimized protocols available, which I will touch upon in a moment. So very briefly, the EZ250 EasySep protocol illustration is very similar to the EasySep protocol I showed you earlier, except that both this protocol and the EZ250 magnet are compatible with a standard T75 flask that allows for that large volume processing. With the EZ250 magnet, users simply add their EasySep reagents to their leukopak sample in the T75 flask, incubate the labeled sample in the magnet for the indicated time, and then pipette off the supernatant to separate the labeled and unlabeled cells. We have also launched larger format reagent kits and optimized protocols to accompany this new magnet for the isolation of a variety of cell types, including T cells, T cell subsets, monocytes, NK cells, B cells, Tregs, as well as PBMCs and depletion of RBCs. The first 12 applications listed here include both optimized protocols and new large format reagent kits, which can process up to 10 billion cells. The PBMC isolation and RBC depletion applications have also been launched with optimized protocols, but they use the existing EasySep reagent formats. If you don't see your specific cell type or kit of interest, please let us know, because we have additional protocols that are in development and would be happy to help you with your large volume cell isolations. Of highlight here, as we are focused on T cells today, I will highlight some of our performance data from our large format negative selection pan T cell kit and our positive selection Treg kit. So to demonstrate the efficiency of the EZ250 magnet, while still maintaining the high purity achieved with the smaller magnetic platforms, here's the performance data for the EZ250 magnet with the large format pan human T cell isolation kit. As shown in the orange bar in the graph on the left, you can isolate T cells from a starting sample of up to 10 billion cells in 20 minutes and still achieve a similar high purity of around 97% as the smaller magnetic platform here that is shown in the light gray bar. We are also very excited to share the most recently launched EZ250 version of our human Treg kit with releasable rapid spheres for the large-scale isolation of FOXP3 positive Treg cells. Shown in the orange bar in the graph on the left, that again performs as well as our smaller magnet platforms that is shown in that light gray bar. This kit also allows for the isolation of the responder cells, which is highlighted with an orange box in the flow plots on the right. And now I wanted to highlight some of our other data for the variety of T cell kits we offer in both negative and positive selection. So this is a snapshot of the data we've generated using the EasySep Magnet. On the left of the screen are the purity results from isolations using our negative selection reagents. And on the right are the purity data obtained using our positive selection reagents. In both figures, the dark gray is the starting purity of the target cell type, and the dark orange bars are the purity of the final isolated fraction using the EasySep Magnet. The purple bars represent comparison data we generated using some of our other magnet platforms, such as the Purple Magnet. For the purposes of this talk, we focused on isolations of T cells using both negative and positive selection approaches. And as you can see, with either method, the EasySep Magnet results in the isolation of highly purified T cells, so either pan-T, CD4 T cells, or CD8 T cells. And the platform performs comparably, or even better in some cases, to our well-established smaller magnet platforms. This consistency in performance across platforms will help to ensure users a smooth transition as they scale up their research. So the EasySep Magnet is a fantastic choice for groups looking to efficiently scale up their manual cell isolations. The protocols are simple and quick, where you can isolate your cell type of choice from a full leukopak in a single magnet in 20 to 40 minutes, while still providing users with the flexibility to further optimize or customize protocols to their specifications. If you are interested in testing this magnet or would like to learn more about it, please reach out to your local sales rep to request a demo. So overall, our EasySep Magnet and reagents are one way to increase efficiency by scaling up cell separation workflows so that scientists can isolate a large number of cells from one or multiple sources in the fewest number of steps. Some other additional benefits of large-scale cell isolation are reduced biological variability of results, as using cells that are isolated from the same donor instead of multiple donor reduces biological variability. In the context of drug discovery and finding therapy candidates, using the same donor ensures that the obtained results are due to the therapy candidate and not due to the biological differences in donors. Another benefit is that fewer donors are required, as isolating a large number of cells from specific donors reduces the need to keep searching for more donors who meet the desired criteria. Lastly, fewer isolations needed when establishing cell banks, as obtaining sufficient cells to bank for future experiments avoids the need to repeat cell isolations later on. Now that our EasySep platform has been out in the field for a few years, we have seen tremendous uptake across a variety of different applications. Here, showcasing some of the applications that researchers have seen tremendous value in performing large-scale isolations. As mentioned is biobanking and large batch cell isolations, as well as immune cell therapy research and genome editing studies, as well as immune response and immunogenicity testing, drug efficiency testing, and screening and immune cell function. This large-scale cell isolation system can accelerate drug discovery and immunotherapy development by enabling researchers to spend less time isolating cells and more time on their important downstream experiments. So while this talk has been focused on T cells, I want to emphasize that we have also optimized this system for isolating a variety of other immune cells as listed here. One that I want to draw attention to that we are really excited about is the CD34 positive selection kit, which is coming soon. This large format kit is optimized for isolation from mobilized leukopaks. And so we also currently offer human mobilized peripheral blood leukopaks, where you can obtain large numbers of single donor CD34 positive hematopoietic stem and progenitor cells from these peripheral blood leukopaks that were mobilized with granulocyte colony stimulating factor, or GCSF, plerixafor, or GCSF and plerixafor as a combo. These fresh mobilized leukopaks are a rich source of circulating CD34 cells and will pair perfectly with the upcoming large format EasySep human CD34 positive selection kit. So stay tuned for more information. With that, I want to bring us back to the workflow and emphasize that while I focused on large-scale cell isolation with our EasySep products, we have products for every step of your immune cell-based research. And later today, you'll hear from Dominika, who will be covering our suite of products supporting activation, editing, expanding, and differentiation, and how they can further support your T-cell research. With that, I want to thank everyone for attending, and I would like to open up the floor for questions.
Fantastic. Thank you very much, Amanda, for that great presentation. I will move now right onto the Q&A and bring you back onto camera, and also bring Fran online as well. Wonderful. Welcome both. Let's jump right in. First question here. We've got a great one. What T75 flasks are compatible with the EZ250 magnet?
Yep, that's a great question. So standard T75 flasks will work. The one that we recommend on our product info sheet is a Corning flask. There are other flasks that we've tested as well that will work. There's a Grainer Bio one that has also been confirmed to work. There's a few considerations when picking a flask. One is the dimensions, so it has to fit properly into the magnet, or the magnetic surface won't be able to efficiently pull the cells to the walls of the flask. Another consideration is how much volume the flask can hold. So there are flasks that can fit, but they can't actually hold the maximum volume that would be able to be used in a cell isolation. Other ones don't work because of the geometry of the opening. So either the angle of the flask opening isn't conducive to be able to put your pipette straight down into the flask without scraping the sides, or the opening itself doesn't easily fit a 25 ml pipette, for example. So ideally, use the one that's written on the product info sheet. If not, you can reach out to your sales rep or product support, and they can help you with knowing which other ones can work too.
Okay, great answer. Thank you very much. Another question here. What are some typical pre-processing steps that you would recommend if using fresh leukopaks?
Yep, that's another great question. For a lot of the products, we recommend a lysis step. So doing an ammonium chloride lysis to remove red blood cells, followed by a few slow spins to get rid of platelets. Often you can also just do a simple wash and go. So if you're looking to save a bit of time and you want to eliminate that red blood cell lysis step, you can just wash the leukopak and then resuspend in the recommended media, which is typically EasySep buffer, so PBS with EDTA and fetal bovine serum, and then just go straight to your isolation. Some specific products will preferentially recommend one processing over the other, so you can, again, look at the product info sheet for the particular product you're looking to use, and it will guide you to how best handle your sample before you get started.
Okay, great. Just a reminder to the audience, if you do have any questions, you can submit them on the right-hand side of the screen now or at any time throughout today's various sessions. Any we don't have time to get to will be sent to our presenters afterwards, and they will follow up, so don't feel free to add them anytime. Another question here, do you have options to isolate low-frequency antigen-specific cells in PBMCs?
Yep, that's a great question. We don't have off-the-shelf products to do that, but what we do have is indirect isolation products. So these are isolations where you can target any antibody as long as it's PE, biotin, or APC labeled. So we have those available in a particle-bound version or a particle-free version, where at the end of the isolation you can remove the particles. We also have a tech bulletin, which I think somebody could probably post into the chat or the handouts, where we describe protocols for isolating either activation-induced markers. So if you want to target your pseudo-antigen specific cells using 41BB, OX40, or CD40 ligand, you can do that. We also have suggested protocols using our indirect system to target tetramers. So you can label your antigen-specific T cells with a tetramer, and then we have a suggested protocol for pulling those out.
Okay, fantastic. Next question here. Can I use frozen cells with the E250 magnet?
That's a great question. I would say, generally speaking, we don't recommend using frozen cells. It's not that you absolutely can't, but when you're dealing with such large volumes and large amounts of reagents that are fairly expensive, we don't want to risk you losing that material, and it's harder to control the quality of a frozen sample. As is often the case with most things, quality in leads to quality out. So you want to make sure if you're going to try a frozen sample that you know that the starting leukopak that was frozen was high viability, in good shape. You want to make sure that you're freezing it in an appropriate way, and you want to make sure your thawing technique is really good so that the cells that come out at the other side are still really viable, are free of clumps, before you even consider doing a frozen isolation. Yeah, so I would say, generally speaking, if you can start with fresh, that is what we would recommend. If you absolutely only have a frozen sample, maybe reach out to your sales rep or product support and make sure that they're guiding you to make sure you retain the highest quality cells that you're putting into your isolation.
Okay, wonderful. And I see Amanda has posted something in the chat, a link in the chat there for reference for everyone. A very quick reminder as well, all of today's sessions are being recorded. You will have access to them and all the resources on demand after the fact. And also you will be automatically redirected to the next session shortly after we conclude here. So stay on the line, as it were. And I think we have time to squeeze in one final question here. What is the typical number of cells in a mobilized leukopak? And is it standard? Is it more than a standard?
Yep. So a standard leukopak is usually in the 10 up to maybe maximum 20 billion cell range. Obviously, that's donor specific. Likewise, mobilized leukopaks do vary. It varies by the mobilizing agent used. So if you use Plerixafor or you use GCSF or you use a combination of the two, it also depends on the actual treatment regimen that's given, the number of doses, the number of time that's passed since the last dose, if it's your first collection or the second collection. So there's a lot of things that affect the actual total number. The number of cells in general, though, that you will get from a mobilized leukopak is more than a standard leukopak. With Plerixafor, you can expect maybe 30 to 70 billion cells. GCSF is usually a smidge less than that, sort of in the 20 to 70 billion range. And the combination, you can get up to 100 billion, but again, there's going to be quite a wide range, but you can definitely expect to get some more cells from those ones.
Okay,
as well,
wonderful.
the cell composition will be slightly different than a standard leukopak.
Okay, great. Unfortunately, I think that is all the time that we have for today. So I'd just like to say thank you very much, Amanda and Fran, for joining us and sharing your insights. It's been a pleasure.
Wonderful. Thank you. Thank you to everyone who attended.